and we can get this show on the road. All right. So the first question is going to be about consensus sequences, and let me share the document so that everyone can see it. All right, cool. All right, so first question, we are trying to find the consensus sequence for this. That's basically all we're doing. So does anybody want to get us started with finding the consensus sequence for this? So let's start with our first letter. So we have A, T, and A. Yes. So everyone says the first one would be A, which is 100% correct. What do we think the second one would be? So Usman says GA and Joseph says R. So the second one would be, oops, I almost unhighlighted too much. So the second one would be R. And so the reason why it's R and not GA is because G and A are both purines. And the way that I remember that is pure as gold. So pure as in purines, and then as is adenine and gold is guanine. And so basically, because those are both purines, that's why we would put in R. And while yes, A and G both show up in equal ratio, so it's 1-to-1, because they're both purines, we would put R instead of GA. That makes sense? I don't know if Dr. Chiari would still mark it as correct if you put GA. I think she'd probably be looking for R. All right, cool. So third letter, we have TATA. Yes, so Joseph's right. R is more specific than GA. GA is too general. And so R would be a better answer. So for the third one, we have AT or TATA. So for this one, we would actually want to put TA because Y's are for pyrimidines. Why was that so hard to say? And so our pyrimidines are G and, or sorry, not G and C, T and C, while our purines are A and G. So if we look at these, A is a purine and T is a pyrimidine. So because they're not both purines or both pyrimidines, we would have to say that this one would be A/T. So does that kind of make more sense now, Usman? When we want to use, okay, cool. All right, so our next letter, we're looking at A, and C. So what do we think that one would be? Yes, so this one would be A because we have majority A's. We have 3 A's and 1 C, so since there's more A's, that's what it would be. Next one, we have A, G, G, G. So what do we think about this one? Yes, G. So, and then next we have G, or sorry, C, G, T, C. So what do we think for that one? Yes, so we would put C because while you have while you have three that show up, 3 different bases that show up, you still have more C's than the other two because you have two C's and you only have one G and one T. So that's why we get C here. And then this next one we have C, T, C, T. So what do we think that one would be? Yep. So for this one, we would put Y because C and T show up in equal amounts and they're both pyrimidines, which is down here. So we would have Y. And while C/T could be correct, Y is much more specific. And so that's the answer that we would be looking for if you were to be asked something like this on an exam. And then the very last one, we have TAAA. Yep. So it'd be A because you have obviously three A's and one T. And so the only thing that I didn't include on here, which maybe I should have, is N. And so the only time that we would have N is if you have an equal number of all of them. So like, let's say this last one, if you actually had TAGC, then this last one would be an N. So maybe I should change that. So this is a better example. Yeah. So because you have one of each of them, they're all showing up in equal ratios. So you would actually have N instead of A like we had before, because before we had three A's, one T, but now we have one of each. So do consensus sequences make more sense to everyone now? I know that when we were going over it in class, it's kind of like, what on earth is going on? So
Question.
yeah.
So you know how for the third one that's TATA, you just wrote A/T.
Mm-hmm.
Then if it was like CA, we would just write the same thing like C/A.
Yes,
Okay.
exactly. Does anybody else have any other questions about consensus sequences? All right, cool. If not, then Garrett, are you still driving or do you want me to keep going?
If you could do one more question, that would be great.
Oh yeah, no problem. All right, next question. So transcription versus translation. This is like something that's a pretty basic question, but like, I guess the two sound pretty similar. And so people tend to get it confused. So we know the central dogma is DNA to RNA to protein. So knowing that, transcription is which part of that? Yeah, transcription is DNA to RNA, and then translation is RNA to protein. Yeah. So this one is like a very basic question, but I notice that people sometimes tend to get it confused. So it's just something to clarify and like make sure you have in your brain. And something that can help if you do keep getting it confused is that transcription has C and translation has an L. And so C comes before L in the alphabet. And so that's how you know you go from DNA to RNA first, and that is transcription. So there are processes that occur during transcription and translation that are common between the two of them. And what is common between the two? And Usman, I'll answer your question after we do this one. Yes, RNA is common between the two, but I was thinking more about the processes that are involved during transcription and translation. But RNA would be correct. This is kind of a vague question. So instead of trying to make you guys like figure it out, I'm just going to give it to you because it is more vague. And so And the processes that are common between the two is that you have initiation, elongation, and then termination. So you're always going to have to initiate somehow, and then you're going to have to elongate, and then you're going to have some way to terminate, because if you cannot terminate, then you're just going to keep going forever and you're never going to get to your finished RNA or your finished protein. So those are like the common processes between the two. And we can also say that RNA is common, because I had not thought about that as being a correct answer, but it very much is. So does anybody have questions about that before I go back to consensus sequences? Okay, I'll take that as a no. And so if not, Usman asked, do consensus sequences start replication, or what is the purpose of it? So basically a consensus sequence is part of the promoter area. And so this is where your polymerase is going to attach onto your DNA or your, onto your DNA. And so because this consensus sequence is part of the promoter area, it's what your polymerase recognizes and goes, hey, like this is where I'm supposed to bind. And then it doesn't actually get transcribed onto your RNA, but it is where you like kind of recognize, and then around 10 base pairs after that is where you actually like start to actively transcribe and start coding. Does that answer your question, Usman? If it doesn't answer, then just let me know and I can talk about it again. But like basically the gist of it is that the consensus sequence is part of the promoter area. So it's like part of the recognition site of your polymerase. Usman, are you still there? All right, well, if not, we can always just come back to it. Oh, you're back. Okay. Did you hear what I said about the consensus sequences or, oh, okay. So basically consensus sequence is part of the promoter area and it's part of where your polymerase will attach. So it's like a recognition site. And it doesn't actually get transcribed into your RNA because it's part of the promoter area. So everything in the promoter area doesn't actually get transcribed into the RNA, but it's just like a place for your polymerase to recognize. Yes, part of the promoter area. Okay, cool. Yes, Joseph. Does the consensus sequence actually exist? Yes, consensus sequences do actually exist. Or wait, are you asking if it's like extremely accurate all the time? The system that we used to find it. That's a good question. I think that this is more so theoretical because of the fact that like we have like over here where we have A/T, we don't know if it's going to be A or T. So like that's more of a theoretical thing. So this is more, it's like a prediction. There's no guarantee that like that's exactly what we're going to find. Yeah, no problem. All right. Does anybody else have any more questions about consensus sequences? Because this is probably something that you will see on the exam or a quiz. All right. If not, Garrett, do you want to take the floor now?
Yep. Thank you. So also my pen stopped working, which is super lame. So I can like go back through my answers in another video so that you like get the extra information you need. But so that's kind of lame. Sorry about it. But we can talk about it anyway. So oh yeah, let me also share my screen. And I also have to present my final project at 1:30 for another class. So we're going to speed run it. Okay. Sick. So genes that are transcriptionally active will select the answer. So in order to answer this question, what Joseph says B. Okay. So transcription, what do we need our DNA to be like in order for transcription to take place? Transcription is like reading a book. In order to read a book, the book has to be open, right? The pages need to be relaxed enough for you to open the book and actually perceive the information that's inside. And similarly, our DNA double helix is wound up very tightly. And in order for it to be able to be transcribed, it needs to be opened up, relaxed. And so I would agree with Joseph's. Yes, exactly. It needs to be a euchromatin. It needs to be relaxed. The reason A is not correct is because if it's open, if it's transcriptionally active, then it is going to be more susceptible to digestion, I guess, because it's open and enzymes can get at the molecules and break them down. B is correct, as we've discussed. It needs to be relaxed in order to be available to RNA polymerase. Having fewer promoters active, that's kind of the opposite of what we need, because we know that promoters are what initiate transcription. And so if we are transcriptionally active, then obviously we are going to have many active promoters instead of few. And having less acetylation of histones, that is also incorrect, because acetylation is what makes euchromatin, is what makes relaxed chromatin, because we have our histones that our DNA wraps around. And remember, DNA is a negatively charged molecule. And what acetylation does is you add acetyl groups to the histones, the histone proteins, and that makes the histones more negative. And because the histones are now more negative, they repel the negatively charged DNA. And when the histones repel the DNA, then the DNA is no longer as tightly wound around the histones, and it is more easily, or it's easier to like get at the information. It becomes more relaxed. So it wouldn't be less acetylated. A transcriptionally active DNA would need to be more acetylated. So the answer is B. Did you do number 3, Sam? I think so.
Yes.
Okay. So did I move on to number 4? Or does anyone have any questions? I think it's pretty intuitive. Easy. Exactly. Okay. Number 4. What does human science know about how to, this is kind of like, I think I gave the wrong impression of what telomeres really are and what they do in the last review. So I kind of wanted to correct that. And it also goes along with transcription a little bit. Not really, but so what does human science know about how telomerase works, and what don't we know? And if I could draw, this would be a much better question to ask. We don't know a lot. That's kind of true for the most part. Humans are kind of dumb. Just kidding. Humans are very capable primates. I will just start by once again showcasing a woman scientist. This is Elizabeth Blackburn. And Elizabeth Blackburn is a German scientist. And she did so much research on telomerase. Her work is the foundation of what we understand about telomerase. And what she found out was that, and we've seen this slide before, but what we found out was that, maybe I should even make it bigger. Oh, I'm also showing the wrong screen. We've seen this slide before. And we know that because right here, oh, you can't see it. Ugh, pain in the ass. So in this top left thing right here, we have missing the primer that's on the bottom part. That's why you can see right here. The primer used to be right here, but then it was removed. And so what we have is this 3' overhang is what it's called. And that's not good. We have DNA sequence that's not attached to anything. So it can't like do its thing as DNA. So what happens is we have this enzyme, telomerase. And telomerase has an RNA template to kind of like buffer up against the 3' overhang. And what it does is that it initiates a new synthesis so that the 3' overhang, this top strand, gets extended. And this is kind of like a moving scaffold, like as these nucleotides get filled in, then it keeps moving this way. And so it's kind of like a moving scaffold that's moving along. And what it does, it extends this 3' overhang on the top strand. That's what we know about telomerase. What we don't know is how the bottom strand comes and gets matched up to this overhanging strand that we elongated. I do like her a lot. She's a cool cat. Excuse me. And, oh, where'd it go? Well, we can put her away for now. But what I failed to mention, what I failed to mention is that telomeres and telomerase, it's not like you have telomeres and they get whittled down and that's really bad. This is supposed to be happening. Do you think the more telomere we have, will it be a problem? And that's where I was going with. Telomere shortening is natural. It's what our cells should be doing. We don't always want to have our telomerase running around and doing stuff, because that's how, like, the shortening of your telomeres is how your cells tell how old they are. And this is important. For example, if I have a cell that recognizes that it's 15, 16 years old, then perhaps that cell is going to start the processes of puberty. And for example, if, so cells knowing how old they are is essential to like your body functioning properly as a macroscopic organism. It's not like a terrible, terrible thing that our telomeres are shortening. Like that's kind of, we need them to for our like long-term growth. And this, like, we think that it's a problem, but it's not. However, it can be a problem if you end up with genetic disorders like Werner syndrome. Someone asked about this in a lecture and no one answered it, so I just wanted to talk about it here. This is a syndrome where telomerase in your meiotic cells, in your gametes, doesn't work. Like it's not upregulated like we talked about in the last review. And so the parent of this woman here, or you can't see her, well, actually, yeah, the parent of this woman that you see, their gametes did not, or excuse me, their germ cells did not properly I'm so sorry, I'm not articulating my speech well. Their parents did not like have good telomerase regulation in their germ cells. And so when they created the gametes that became this woman was born with prematurely shortened telomeres. And so, like, as soon as she was born, her body thought it was older than it actually was. And so, probably we can see that at age 15, she's appears relatively normal for that age. But then, only at 50 years, this woman looks like she's 90 years old. That's because somewhere probably in her 20s or something is when the effects of her prematurely shortened telomeres began showing their effect. And so she experienced all the symptoms, with shortened telomeres. Her cells thought they were really old. So they made skin that was less elastic. There's less molecular machinery going around that is like cleaning out your arteries. So you have a lot of blockage and heart problems and stuff like that. So yeah, I just wanted to explore that a little bit. And so what we know about telomerase is that it extends the 3' overhang, and we don't know how it makes the complementary sequence to like fill in that overhang. So it doesn't overhang. People with cancer are more at risk of this. People with cancer are much less at risk of this, because what this woman experienced was prematurely shortened telomeres because her parents' germ cells did not have a proper amount of telomerase. They had too little of an amount. But cancerous cells highly upregulate telomerase. So their telomeres are perfectly long, able to like pretty much be immortal. Oh, did you want me to keep going?
Oh, sorry. I didn't see that you stopped sharing your screen.
No.
All right. So our next question is basically just talking about upstream and downstream of a promoter. So if we have -35 and -10, would these be upstream or downstream of the promoter? Yes, so they would both be upstream, because anything that's negative is upstream, like Usman said. So both of these would be upstream. And so 2 and 20 would be upstream or downstream. Yeah, downstream, because positive is downstream. And so what is significant about what is this number related to the 35 and 10 and 2 and 20? 2 and 20 were just like made up numbers that I made up. -10 and -35 do have significance to us. And so my question to you guys is, what's significant about the -10? Why should we even care about it? Yes, it's the Pribnow box in prokaryotes. And so what does the Pribnow box tell us? What's the point of it? Yes, so it's where we attach our polymerase. And so I kind of talked about it before. But here's a better, more concise explanation. So basically, this is where your RNA polymerase is going to bind and you're going to start transcription. But like I said before, these nucleotides don't get transcribed. And so, okay, so I'll answer Carla's question first because much shorter answer. So is it always going to be the number that's negative? So yes, your pribnow box is always going to be at -10. So it's always going to be upstream from your promoter because you want to make sure that you attach before you actually need to start transcribing. Because if you were to start transcribing at +10, then you're going to miss the first 10 nucleotides that were supposed to get transcribed. So Joseph asks, is the Tata box in eukaryotes at -10 or is it somewhere else? I'm glad that you mentioned the Tata box because I was actually going to ask that. I was going to ask what was the analogous structure in eukaryotes. And so it's the Tata box. And I am actually not sure where it's located on eukaryotes. I can look it up though and let you know. So no, it's not located at, or wait. Usman, are you talking about the Tata box or the Pribnow box? Oh yes, Usman is talking about the Tata box. So yeah, according to Wikipedia, which I don't know, do we trust it? Do we not? Who's to say? But it says that the Tata box is normally 25 to 35 base pairs upstream of the transcription start site. So hopefully that answers your question, Joseph. Okay, cool. So does anybody have any questions about that? This part is pretty self-explanatory, but I just wanted to make sure that you guys understood it because sometimes like the upstream downstream lingo can be confusing. What's present at -10 in eukaryotes? I actually have no idea. Probably just mumbo jumbo nonsense. Or like not nonsense, but like stuff that doesn't get transcribed into your RNA. Yeah, I could
part
be
of the
part of the
consensus maybe.
All right, cool. I will stop sharing then. And Garrett, you can take it away.
Sure. Okay. Oh, and if you ever are confused about the upstream downstream thing, think of it like, I guess, like say you have a number line and you have 0, then you have negative numbers and positive numbers. And so if 0 is like the start of transcriptions or transcription start site, you're going to be transcribing the positive numbers, and the negative numbers are going to be where you have your regulatory elements. And we can also think of it like a stream, as the lingo would imply, that usually we read from negative to positive numbers. And so like it's going to flow this way. And usually rivers flow from upstream to downstream. So positive numbers are further downstream, pretty intuitive. But anyway, if you could please label the colored sections of this mRNA fragment. So what are these dark bluish, or excuse me, these, oh, you can't even see my thing. Ugh. What is this color? What's this all about? Usman says non-coding region. Interesting. Or coding. Which one? You have to decide. It is present in the beginning too. Yes, so when we transition from this to this is our pre-mRNA, and this is our, what's that called? Like manufactured. There's a better word for that. So I will just come out and say that these dark turquoise areas, these are exons. These are exon or coding fragments of DNA. What about this and this? These are our they're called intron sequences. These are our exons, or the dark color, the light color are introns. And then what happens is that somehow this pre-mRNA sequence gets changed into this mature mRNA sequence. And what is that process called? It is not transcription. Because we have mRNA here, and what we're doing is we're I shouldn't use trans, we're altering it. No. I'll give you a hint. This process is completed by the spliceosomes. Splicing, exactly. This process of changing our pre-mRNA, the top bar, is what we get out of transcription. And so we need to prepare this for translation, right? Oh, well, I guess I gave away the second part of the answer. This happens after transcription and before translation. We need to prepare this top DNA strand that comes out of transcription so that it can be used in translation. And so what we do is we put this yellow thing on the front and this dark blue thing at the end. What is this yellow thing? And what is this dark blue thing? We know that these are all of our exon or coding sequences that create our codons. So Usman thinks that this yellow thing is the promoter. Do we have any other ideas? We learned about this in Bio 213. Well, I personally don't have a lot of time, sorry. So I will just come out and say that this is our 5'. Oh, that's a bad 5' G cap. This is a bunch of sequences that are full of a lot of guanines, and it's kind of a promoter. This will go into our translation, and this G cap signifies that this is the beginning of our mRNA sequence, and we are ready for this to be translated into amino acid polypeptide chains. So what is this dark blue? Yes, this is our poly A tail. This is a bunch of adenines all together, and it serves kind of a similar function to telomeres on our chromosomes, because we have all these A's that are just, not doing too much. And when this molecule is bumping around, doing stuff, it can get damaged. And it commonly gets damaged here at the end. So if we fill the end with a bunch of stuff that we don't need, we can get it damaged, and it's not going to harm any of the important exon coding sequences that we use. And like I said, this is called splicing. And it happens after translation, or excuse me, after transcription, before translation. Any questions? We only touched on it a little bit today, and we kind of didn't give it its due diligence, but we did learn about it in 213, so it should pretty much just be a review. Yeah, someone can share the secret question. So if there are no questions about number 6, let's move on to number 7. This one's a bit more involved. So we have this here DNA sequence. Scientists study the products made by this sequence in 1,000 different cells, and they find that 300 of them produce this arrangement of polypeptides, 450 produce this arrangement of polypeptides, 150 have this, and 100 have this. How could this be the case that this same DNA sequence is producing different arrangements of products in different cells, even though it's the same sequence? How could this be happening? Sorry, chat's not opening up. Joseph says it depends on the splicing. Yes. Yes, Usman, good job. Alternation. Oh, which one? You have to choose, Usman. Is it alternation or multiple splicing? No, you cannot say no. Oh, I wish I could draw this. This would be so... Okay, I will talk more about this when I can draw it. The answer is multiple splicing. And let me pull up the slide since I can't draw it. I don't even have these ones downloaded. So my password. Okay, wait, we have either alternate splicing and multiple 3' cleavage sites. So which is it? Wait, we have either alternate splicing and multiple splicing cleavage sites. So which is it? Hold on, let me pull it up. Yeah, this is what we learned today in class. I told you I'd have a question about it. Where are we? This is chapter 14, right? Okay, thank you. Excuse me, sir. Excuse me, sir. Okay, here it is. It's on slide 24. Yeah. Slide 24, if you look at the right side, you see the multiple cleavage sites. And having multiple cleavage sites is, for example, if we had this sequence of DNA here, what we have is obviously our start codon here at the beginning and our stop codon here at the end. But these 4 sequences in the center, these 4 codons, because you see they're all arranged in groups of 3, which will go into the tRNA. Yeah, so if we have multiple cleavage sites, let's say that we have a cleavage site here and a cleavage site here. But then we have another cleavage site in a different, maybe in this cell you have the blue cleavage sites, but in a different kind of cell you have, oh, I didn't even change the color. I wish I could draw this. So whether or not the cleavage site is here versus here versus here versus here will like determine which exons get stuck together. And that, as we can see by this codon table, will produce different amino acids. Yes, multiple cleavage sites, because we have multiple cleavage sites. Instead of just in between the first codon and the second codon and the third codon, we can have it in between the first codon and the fourth codon, in between the second and third codon and the first and second codon. So having different, oh, I just realized, sorry, I'm using two different computers at once and I'm not paying attention to. I thought we can get different products from alternate splicing too, because we're removing some parts of exons. You can get different arrangements of codons. Oh my God, Joseph, you're, oh, I blundered that whole question. So yes, damn, I wrote the wrong question. So what I was trying to get at was that like, if you had a splicing event here, or if you had a cleavage site here and here, for example, this would produce like these codons. But if you had a cleavage site, or actually, let me restate that. If you had a cleavage site here and here, then obviously you'd get CTT, and this would become the anticodon GAA, which would produce glutamine. But let's say instead of having the cleavage site at there, what if you had the cleavage site here? And instead of having CTT, you'd have TTC, and that no longer produces glutamine. That produces AAG, which is lysine. So yeah, I thought I was making one question, but I accidentally made another. So this occurs from alternate splicing because it splices in differing or altering places within your coding regions. So if you had splicing here at this region versus at, how do you change the color? At this region, you would get different products. Oh my God, I thought I changed it. This isn't going my way. Yeah, I'm sure you can get the point. And I will redo this and make sure not to give you the wrong answer. So thank you for that, Joseph. So yes, this would be alternate splicing. That's how this happens, where you can have one single gene sequence producing several different products. Yeah, in general, you can. That's the whole point of alternate and multiple splicing events. That's how you can have, how will you know which one it is? You will be given a question that is more well thought out and it will like elaborate on this sequence and how, well, I guess you guys have used codon tables, right? I keep forgetting. I'm going to zoom out. Yeah. So this DNA sequence, TAC, which RNA codon sequence is it going to correspond to? Yeah, it's AUG, because, you know, T pairs with A pairs with U, C pairs with G. And so you're looking for AUG, so you can find your way down to the table, and you have A as your first base. You're looking for your second base is U, so we know we like triangulate. Oh, I forgot, you can't see my cursor. Oh my God. So we have AUG is what we're looking for. And so we look at our first base is A. Then we have U, so we find U as our second base. So we kind of triangulated, and then we're looking for G. So we can look at this square for our AUG, and we found it right here. It is methionine, which is the start codon. That's a good way to tell if you are doing it right, is that you should usually start with the start. And then you look at this sequence here. No, not that one. Grr, whatever. The TGA, and you do the opposite for that. You transcribe it from DNA to RNA. So this would be ACU. And we have ACU, which is threonine. And so you can compare the DNA sequence using the codon table to the corresponding amino acid. And that's how you can check. I think that's a question someone asked. So I will definitely not mess up this question in the future. Thank you for correcting me. And yeah, you just work backward. So that is all Sam and I had. Actually, did Sam have another question? No, I think we're good.
No, I didn't have any more questions.
Well, I am going to firstly, I'm going to explain it better later, but just like Joseph said, you have to look at the products that you see here, compare them to the codons you see in the codon table, and then see and draw in yourself where you think the cleavage sites would be based on the products that are created. So I gave you an extra challenge in giving you the DNA sequence instead of the mRNA sequence. So you just have to transcribe it first, and then you can directly compare it. It is almost 1:30, so I'm going to scamper away. I don't think
Wait, Garrett, before you go, I'm gonna stop recording.